First, welcome everyone to the last Teen Science Cafe of the school year. Congratulations to all of our graduating seniors and for the rest of y'all for getting another school year under your belt. So last week, in keeping with the Teen Science Cafe's policy of anti-racism, we provided a series of resources for you to peruse and ponder. We know things are still hard out there for Black folks and people of color around the world, and that movements take time. Morehead Planetarium and the Teen Science Cafe want to share some more resources to help keep momentum going with the goal of seeing real systemic change. Change happens with help from all of us, so I'll be adding them to the chat in just a few minutes to read and watch at your leisure. And again, thank you all for joining us. And now I'll cede the screen to our moderator, Riley, so she can introduce our speaker.
Okay, so this week we have Megan Damico, who is a third-year PhD candidate in environmental health sciences at UNC Greensboro. She studies how different factors like diet and age can influence the composition of insect gut microbiomes and uses honeybees as a model organism to answer these questions. Megan loves birdwatching, reading, and hiking in the Appalachian Mountains. And without further ado, I'll pass it on to Megan.
Thanks, Riley. Awesome. Let me share my screen. All right, hopefully everybody can see that all right. Thank you so much for that introduction. I am so happy to be here with all of you today, and a huge thank you to Morehead Planetarium for inviting me to speak. So yes, my talk name is Bugs and Bugs, and we're going to be exploring the incredible world of insect gut microbiomes today and the world of microbial communities. And if you're really interested in learning more about what my research is and kind of what I do as a scientist, feel free to follow me on Twitter. Post a lot of cool information and random stuff there too. But let's get started. So first and foremost, I'll introduce myself and kind of talk about my career goals and my academic timeline, really how I got here today and what that means to me as a scientist. And then we're going to jump right into the incredible world of microbial communities and how important they are for us as humans. And then lastly, we'll talk most importantly about the honeybees and their gut microbes and why they're also a really important part to honeybee health overall. And feel free to input questions to the Q&A box. I'm really curious to see what you all know or what you're curious to learn more about. And I'm very open to answering all the questions you have. All right, but first and foremost, who am I? Why am I here? My name is Megan Damico, as Riley said, and I'm a first-generation college student from Livonia, Michigan. You're familiar with the Michigan area that's just outside of Detroit. And I got my bachelor's degree of biomedical sciences and microbiology from Grand Valley State University. So that's just on the other side of the state of Michigan. So currently, I am a third-year PhD candidate at UNC Greensboro in the biology department, and I am studying honeybees and their gut microbes. But I wasn't always a scientist, and if you don't find me in my lab, I'm definitely going to be outside bird watching or just kind of relaxing and reading a book. I really enjoy being outside even when the weather is kind of what it looks like today. So to kind of walk through what my academic timeline looks like and what my current career goals are, because I'm still a student, I wanted to kind of walk through this timeline and see if you maybe have any relations to me in terms of I felt the same way or those sound really interesting. I'm also open to answering questions about what kind of it might look like to be in college and study science too. So first and foremost, I graduated high school in 2014. And coming out of high school as a first-gen student, it was really interesting to see how I was going to navigate college. For me, first generation means that I don't have any immediate family members or distant family members who've ever gone to college before or have some sort of college degree. So navigating the college environment was really difficult for me at first. I didn't really know how it worked. I kind of was just told, go to class, make sure you eat food, go to the cafeterias, etc. So it was kind of like a crash course. And it took a long time for me to get used to that. But coming out of high school, I hated science. I absolutely hated it. I didn't have teachers who were really supportive. I also just was constantly told in those science classes that I did get to take that I wasn't good enough. I didn't score the highest in the exams that I took, and I wasn't really interested in some of the content they were teaching. The only class in high school that made me go into college to study science was my AP biology teacher. And that class I took on a whim. I was like, I'm going to challenge myself. People keep telling me I'm stupid, then I really want to prove it to myself that I'm not stupid. And I was interested in the subject. And it turns out that class was the entire thing that changed my career. I loved biology. I was good at it. And so I convinced myself at the time that I wanted to be a medical doctor immediately. It's like the first thing you think about when you're going to college is I'm going to be a doctor, a lawyer, or a doctor lawyer, or a combination of another career that makes your parents really proud, I guess. So I went into college thinking I was going to be a medical doctor. Boy, was I wrong. I definitely learned along the way that that was not what I wanted to do. And that took a lot of time to think on those things. And so as I got into these college courses, I kind of realized that I didn't really like the competition amongst other students. I wanted us to all work together towards a common goal, but there's a tough competition to get into medical school, and other students were really focused on that, and that was great, but it just wasn't for me. And I really wanted to find something that sparked my interest a little bit more. So I started looking around into student groups just to try and see what I was interested in. And the first group I found was called the GVSU Beekeepers. And I know that sounds really strange coming from the middle of a school that's kind of surrounded by agriculture, but there were a group of students who were really into bees, and it turns out that I was also one of those students. And I grew up in the suburbs, didn't really have any experience with bees, but over time I ended up just really, really loving it. And that's kind of where it all began, I guess you could say. And so there were a few, there are a few highlighting experiences when I kind of got out of college that helped get me to where I am today. And those were really internships that I applied for and having mentors who really helped me along the way. And so throughout college, I thought I was going to be a farmer or a beekeeper, and then I had no idea what I was going to do. And then it really came to finding my place in science and knowing that one class could change my outlook on science in high school. And the same thing happened for me in college. I entered into a microbiology course in my junior year, and I realized that that was the science for me. That's kind of all it took, was to find a niche in science that I loved. I really, really enjoyed learning about microbes and how they could cause harm, but they could also be extremely beneficial for us. And so with that kind of passion for microbiology and also these honeybees, I applied for an REU program at UT Austin and got accepted to go do research in a lab that studied the honeybee gut microbiome. So my whole kind of research career really started in honeybees in my junior to senior year kind of summertime. And then as I came back from that internship and started my senior year, I applied to graduate schools and ended up getting a position here at UNC Greensboro. And so I wanted to be a college professor in that time, kind of moving forward. And I started my PhD very shortly after I graduated. I have not been out of school since I started kindergarten, and it's been a really long time. And so now that I'm in my PhD and kind of learning about all of these different things, I'm realizing that bench science maybe isn't directly what I want to do. I really want my research to have kind of a broader impact, not just to the researchers in my field, but to everybody else in the community that I live in. And so kind of throughout my time as a graduate student, I've thought about becoming a data scientist or being a full-time science communicator. But what's really sticking and resonating with me right now is actually merging two things that I really enjoy, which is politics and kind of this idea of advocacy for science, and using my technical expertise as a scientist to help policymakers make better policy for our communities. And that's kind of the field of what science policy really is. And so looking forward to when I graduate in 2023 with my PhD, I'm really trying to see the outlook on how I can be a better scientist as a science policy analyst. And so really what this all means, as I blabber on continuously about myself here, is that I want you to explore your interests and know that your interests are going to change, but they're also going to help shape what your career might look like, and to follow those passions that you have and really let them take you into really interesting places. So just a few photos I wanted to share. I had a different internship in college that I was an urban beekeeper in the city of Chicago for the Chicago Botanic Garden. And so this is me on top of the McCormick Center keeping bees. It's a huge conference center. And then these are some of my fellow scientists that I had during my internship program that I was roommates with. And so they're all engineers, and they taught me a lot of weird stuff that I don't remember, but it was an awesome experience, and I highly recommend those places. So let's jump into the science. I don't know if we have any questions, if we want to pause for a moment. If not, I'll keep going. Okay. Awesome. So first things first, let's start with a poll. You guys should be getting that pretty soon. And the question is, how much weight do bacterial cells contribute to your body? Answer A, 0.4 pounds, B, 3 pounds, C, 10 pounds, D, 50 pounds, or E, bacteria are too small to have any weight. What do you guys think? All right, results are in. So how much weight do bacterial cells contribute to your body? 67% of you said 0.4 pounds, 0% say 3, 33% say 10 pounds, 0% say 50, and nobody said bacteria too small. Very good. I'm incredibly happy to hear that nobody said E. So the correct answer is A, 0.4 pounds. So crazy enough, bacteria actually contribute around 2 kilograms to the overall human body weight. And 2 kilograms obviously estimates around 0.4 pounds. But what's crazy is that just in the colon alone, which I have pictured here on the right, bottom right-hand corner, have around 10 to the 11th bacterial cells. It's 100 billion bacteria cells in your colon alone. And not to think that there are also 10 to the 11th bacterial cells on your skin, and in your mouth. So we are covered in bacteria all the time, every single day, really meaning that you're never alone. So if that comforts you or makes you uncomfortable, I'm sorry, but it's kind of a fact. So now I want to talk a little bit more about why we'd want to study microbial communities. So I have these two sorts of communities in comparison here, and I want to kind of draw some similarities and some differences between the two. So if we think about mammalian communities, say like cheetahs in the Serengeti of Africa, we are able to look at their food patterns, what they like to hunt, at what time of day, what behaviors they might have, and what interactions they can have with other animals that share that ecosystem. We're also able to look at maybe where they nest, their reproductive habits, and their den locations. So clearly, this is how we've been able to look at many different organisms, and through many years of ecological research, theories have been formed to show how these communities interact with one another. Well, obviously, being microbial communities, we can't really do that because it's hard to see microbes by the naked eye, so we have to use other resources. We can still do really similar analyses on these microbial communities, like look at what food types they're eating, so like micronutrients, think like the iron, zinc, and carbohydrates from the food that you're going to be eating. They're also going to be moving around quite frequently. Bacteria often have these appendages from their cells called flagella, which are these long appendages on the backside, or cilia, which are these hair-like particles kind of coming off of their sides. So that helps them move around in the environment a little bit. They're also going to have, I guess if you could say, nests or very specific places that they're going to inhabit. So it depends on the environment that that microbe likes. Maybe it needs a really low basic pH or a low acidic pH or a higher basic pH, or it just likes this specific location in your gut. So they're very specific and have kind of picky habitats that they like. But a lot of the terms that I'm going to be throwing around this talk, I want to make sure that we're really solid and have a good understanding of. So first and foremost, microbiome. I've already said it a few times, but what does that mean? A microbiome is simply just a collection of bacteria and their genetic information in an environmental habitat. So whether that be a human gut or the soil, there could be a collection of bacteria there living in some sort of symbiotic relationship together. We also have different types of microbiomes. So for example, the ones that I'm focusing on today in my talk are host-associated gut microbiomes. So these are going to be microbiomes that are associated with a host and located inside of a gut of an organism, whether that be a honeybee or a human or what have you. And so a lot of the times when we think about host-associated microbiomes, we think about these symbiotic relationships that they might have with their host. So if you've heard about symbiosis before, maybe in a biology class or maybe casual talk with your friends about symbiosis, that'd be pretty cool. There's 3 different types. There are mutualistic, parasitic, and commensal. So a lot of the times we think about parasitic because they're kind of creepy. They creep me creep me out at least. So parasitic is a relationship between a host and maybe an organism that's inhabiting that host. And one organism benefits and the other organism does not, has a negative benefit or a negative result from that relationship, I should say. The other symbiotic relationship could be mutualistic, where both organisms are benefiting from being with each other. And then lastly, there's commensal, where one organism benefits and the other organism neither gains nor loses something from that relationship. So just keep these definitions in mind as I go through. Oftentimes I exchange microbiome for microbial community. So if you get confused, feel free to drop a question in the question and answer box and I'll clear that up for you. Okay, but most importantly, if you take anything away from my talk today, I want you to know that microbial communities are really important for many different organisms. Life on Earth is sustained by these microbial communities, and they're not just bacteria. I mean, to think about archaea, viruses, fungi, algae, and protists, they're everywhere. And they're all existing inside of these tiny microscopic communities that we never think about. Specifically for gut microbiomes, or for just microbiomes in general, I should say, they're really vital for environmental processes like the carbon and nitrogen cycles that exist in the soils, plant growth and productivity, applied microbial processes, which I don't really think about that much, but it's true, thinking about biofuels and waste treatment, as well as animal health and nutrition and development, which is going to be really important for my talk today. So on the top left-hand corner, I just wanted to highlight many different organisms that have gut microbiomes in specific, going from termites to ticks and honeybees to zebrafish, all the way around the circle into your cats, dogs, and humans. And those are host-associated gut microbiomes. Well, we also, again, have microbiomes that exist freely inside of the environment, such as in the ocean next to these geohydrothermal vents. It's pretty incredible. So some of you might be wondering, okay, Megan, this is great. We know that microbes are important. They exist everywhere, blah, blah, blah. But how do we actually see them? How do we know what they're doing? And we owe this all to incredible technology called genome sequencing. And so what that means is that we're able to use technologies to tease apart the G, T, A's, and C's of a genetic, of the genome of an organism. So we're able to tease that apart with this technology to actually see what the genetic recipe looks like for that organism and what it's making, what types of proteins. Is it upregulating this protein versus downregulating another? And so what this timeline is kind of showing you is when microbes first were isolated, when DNA was discovered by Rosalind Franklin, Watson, and Crick, when we saw DNA sequencing actually being developed, and kind of continuing on since this technology has been developed. We're able to see that the human genome was able to be sequenced in 2003. And since then, we've found more technologies that make sequencing any sorts of DNA quicker, much cheaper, and way more accessible for the floodgates of research to be opened up into the world to see which bacteria are living anywhere. We can kind of figure that out now, which is really cool. And so the process by which I study gut microbiomes is what I call community profiling. And so oftentimes in gut microbiomes, we're looking at poop. It kind of stinks, literally and figuratively sometimes, but it's worth it. I get to see what microbes are living inside of an organism's gut, and I think it's all the best. So I can, say, extract microbes from the poop of an organism. I can then take that DNA that I've extracted from that poop. I can put it into a sequencing machine, which will then spit out the genetic code of those bacteria, for example, living inside of that gut microbiome, excuse me. And then I can start to tease apart using computational techniques to figure out which bacteria are actually living inside of that organism's gut. And so that's what I call community profiling. All right, let's keep going. So another question you might be thinking of is, okay, so we're talking a lot about different organisms that have these gut microbiomes and how important they are. So why don't we just study that in humans? Well, it definitely has been studied in humans, but there's a lot of challenges that come with that. So first and foremost, the human gut microbiome is incredibly complex. We as humans are host to trillions of microbial cells in each individual person, and a lot of those bacterial cells are inside of our guts. But on top of that, humans are home to hundreds of thousands of species of bacteria in that gut community, making it really difficult to tease them apart using computational techniques. It's really hard. Another issue is that we don't really have a clear answer as to what a healthy or a stable microbiome looks like. We don't know if one person who works out 6 days a week and only eats kale and smoothies is extremely healthy in their gut microbiome, or if a person who eats bacon 3 times a day is having a healthy microbiome. There's not a clear answer on that, so it makes it tough to know what a normal gut community looks like, especially in humans. And lastly, you can't experiment on humans. It's not ethical. Again, you can't make somebody eat kale only every single day, and again, somebody eat bacon every single day. It doesn't really work like that. And so in order for us to really understand what these communities are doing, what are they digesting, what are their interactions with other bacteria, we need a really simple model system is what we call it, a simple organism that has a very small gut community that we can manipulate easily and is not going to have terrible ethical concerns when we're trying to do experiments on them. So in this case, that is why I use the honeybee to understand what gut microbiomes are actually doing. And so from there, I'm gonna bring us to the incredible world of honeybee guts and why we study them. Okay, so in order to really understand why honeybees are awesome and really great for studying the gut microbiome, we first have to kind of tease apart what goes on inside of a honeybee colony. So first and foremost, there are 3 types of bees that you'll find in a colony. You'll find a drone bee, which are the only male bees that live inside of the colony. They are around 0 to 500 of them during the spring and the summer. And as you can see from this picture here on the right, they have really, really big bug eyes. And they're kind of creepy to look at when you're in the colony. Their only job is to mate with the queen in the springtime, and they actually die after mating, and it has an audible popping sound when you can hear them mating and then dying. It's really scary. But they live for around 57 days, and they do play an important role to introduce genetic diversity into the colony and make sure that the colony can survive, obviously inseminating the queen. So that leads us to the queen bee. Queen bee in this case is not Beyonce, although I wish I had Beyonce beekeeping with me every single day. That would be incredible. The queen bee is the only fertile female inside of the colony. As you can see here, she has a longer abdomen than the drone bee, and her thorax, as we call this really hard darkened part here, is going to be a little bit less hairy than some of the other bees in the colony. She's going to live for around 3 years and produce almost 2,000 eggs per day. That is her only job, to lay eggs and consistently populate the colony. And lastly, we have the worker bees, and potentially the most important for my research because I only use worker bees for my experiments. Worker bees are the workforce of the colony. They are non-reproductive, and a healthy colony can have up to, if not more, 60,000 worker bees at a time. I would even say probably a really healthy colony as of right now in North Carolina would have around 80,000 bees because it's nice and warm and they are definitely out foraging. So they live around 4 weeks during the active summer seasons, and then when they go into hibernation in the wintertime, they're going to survive for probably 4 to 6 months, depending on where we are geographically. So for my research, like I mentioned, we only use worker bees to study their gut microbiome because it's very consistent, whereas the other 2 bee types in the hive have a less consistent gut microbiome community, and therefore we don't use them as often. So some of you also might be wondering, well, how do they actually get their microbiome? How do they get those bacteria from one place to another? And it's a pretty cool process. As you can kind of see from this timeline here, as worker bees, you have bacteria in the gut on the y-axis and just time and the different developmental stage that the bees are in. You can see that they're going to shed their gut lining at like day 10, which is, you know, great. And then as they emerge as adults, what happens is that honeybees are going to be constantly having social interactions with each other. So these different types of social interactions are where they're going to get the gut bacteria for their gut microbiome. And that's mainly going to be through fecal oral transfer, where they're going to eat tiny bits of each other's poop. Again, we're talking a lot about poop in this talk, but it's really important to know that many organisms do this, and it's important to get that gut bacteria. So they're going to be doing this predominantly through fecal oral transfer, but they can also get gut bacteria from the hive materials or through what we call oral trophallaxis, where they're sharing food between each other. So the honeybee gut microbiome, obviously bees are really important agricultural pollinators. So by using them as a model to understand what their gut microbiomes look like, we're also answering really important questions about how we can keep them healthy and strong and improving their populations for the future. As I just mentioned, they're going to socially obtain their gut microbiome through their sister bees. And the species of bacteria found in the honeybee gut are only found in the bee gut, meaning that you couldn't go out and sample soil, sequence that soil, the DNA of the bacteria, and find bee gut species. So they're really, really exclusive to the bee gut. And also important is that the gut microbiome of the bee is really simple, and we can culture all of those species inside of my lab, and you can actually raise them without their gut bacteria. So if you remember before when I was saying we need model systems that are really easy to manipulate, well, what happens when a bee doesn't have its gut microbiome? Does it survive? We know that now because we're actually able to manipulate it. And so what I'm showing you here on the right-hand side, on the y-axis here are just a list of really common microbiome model systems that are used in the field, and then some characteristics that are often needed when looking at these models. And so all I want you to take away from this is that the lighter color, the circle here, I should say the more white the circle is, the more that model meets the characteristics on the x-axis. So basically, the honeybee wins. It kicks everyone else out of the league. It's great. I love the honeybee for that reason. All right, looking at the structure now, like actually looking at who's in the gut microbiome and why is that important. So there are 8 total species of bacteria inside of the bee gut. And again, if you think back to humans where we have hundreds of thousands of species, no wonder we don't use humans. It's way too complicated and there are so many other issues that we need to think about. But for honeybees, there's only 8 total species. And actually 5 of those 8 species are what we call core members of the bee gut. So in this figure here on the right-hand side, I'm kind of showing you, what the bee gut looks like. So we've got their mouth and where they store honey, kind of moving along into what we call the ileum, where 2 of the core members live. And then the rest of the bee gut bacteria live in the rectum. And so they're very spatially organized. The honeybee is extremely specific about where its bacteria lives. And these bee gut species, these bee gut bacterial species are really stable across individuals and populations. So if we sampled a bee gut microbiome in Durham, and then we sampled a bee gut microbiome in China, their guts would have the same bacteria and at really similar abundances too, which is crazy to me. I think that's awesome. So we know that the gut microbiome is important for bee health and many other organisms too. Their gut microbes are great for their health and development. And through previous research on the honeybee and its gut microbes, it's been shown to play a role in boosting the immune system and also playing a role in the metabolism for the honeybee when it's breaking down the pollen and nectar that it has, that it collects in the environment. It also is important for hormonal control and the behavior of the honeybee, as well as growth and development. And so what this figure is really showing you here on the right is that many different factors can alter the gut microbiome or make it not normal, is what we would call it. And so when that happens, the possible consequences for the honeybee are that all of these different effects that I just mentioned could make the fitness or the reproductive outcome for the honeybee be lessened or impacted a little bit. So also another thing to note is that the honeybee microbiome is really important in making sure that pathogens don't come in and take over that gut community, which could also make honeybees pretty sick as well. All right, and this is my second poll. Which type of bee is responsible for making honey? Is it A, the queen bee, B, the worker bee, C, the drone bee, or D, the honeybee? What do we think? I feel like I should have Jeopardy music playing. Okay, awesome. So the poll results are in. So which type of bee is responsible for making honey? None of you said queen bee, which is good. All right, 80% say worker bee, very good. None of you said drone bee, and then 20% of you said honeybee. All right, so the answer is worker bee. And I tricked you. If you said honeybee, you're not wrong. but we want it to be a little bit more specific. So worker bee is correct. Worker bees are the ones that are doing all of the work in the colony. They're the ones who are most populous, so having like 60,000 workers in a colony. A single colony alone can make up to 111 pounds of honey each year. And in fact, in my personal colonies that I had in Michigan, I extracted 300 pounds of honey from my colonies one season. It's a lot of honey, it's a lot. So a single bee must drink from over 1,000 flowers in order to fill its crop or its honey stomach with nectar to only make 1/12 of a teaspoon of honey in her lifetime. And so that's a lot of work, a lot of foraging time. And I just want to show you here kind of what a honey frame looks like inside of a bee colony. So you can see all of our worker bees here on this frame, and all of this kind of waxy stuff here, that's honey. So the bees have just capped it to make sure that it's going to have a long shelf life, and everything that's uncapped is still nectar that they're going to be turning into honey very soon. Okay, poll number 3. How do honeybees contract their gut microbiome? Is it A, drinking honey from the hive, B, foraging from flowers, C, social interactions with other honeybees, or D, taking bee probiotics? What do you think? All right, so it looks like it's a pretty unanimous vote here for C, which is social interactions with other honeybees. Great job, everybody. Awesome. So yes, indeed, honeybees will contract their gut microbiome through social interactions with other honeybees. And I really love this picture here because this is a bee that's just emerging from her cell. She's a baby bee. And you can see directly she's sticking out her tongue, or what we call a proboscis, to another honeybee that's also going to be giving her food or gut microbes in this case. So great job, everybody. Okay. So a lot of what you might have heard on the news or just in general is what is causing the decline of honeybees. In recent years, we have seen dramatic numbers of honeybee populations in decline, and this is due to numerous different factors. So we have an increase in disease prevalence from viruses to bacterial infections. We have an increased use of agricultural chemicals. We also have a lot of habitat fragmentation or just habitat loss in general. But we can also add to that list microbiome disturbances. And so that's what I'm going to be talking to a little bit in just a moment. But what a lot of research is trying to understand are how do these agricultural chemicals impact the honeybee gut microbiome as we kind of move from this forested habitat on the plainlands to a lot of these monocrops or really big monocultures of corn and soybean, etc. And so some of these agricultural chemicals include pesticides, fungicides, herbicides, but if you didn't know, antibiotics also play a role in this too. And so we talked about the good bacteria that are doing a lot of awesome things for honeybees, but there's also some bad bacteria that exist too. Some major bacterial pathogens that honeybees can actually come in contact with are European foul brood is what we call it, American foul brood, and both of these are going to only impact baby bees inside of the colony. Or we have this thing called idiopathic brood disease syndrome, and it's also known to impact the larvae. So all of these two specifically here are bacterial infections, but as I mentioned here, the causative agent is unknown, meaning we don't know which bacteria is actually causing this infection. So these bacterial infections are treated with antibiotics, terramycin and Tylen. Those are going to be distributed by vets to beekeepers to make sure that their bees are okay. And just an example of what healthy larvae for bees look like, you can kind of see these tiny little squishy things in here inside of the cells. This is what a good brood pattern or a good laying pattern would be like for a queen. Happy, happy. And then we have a poor brood pattern. And poor brood pattern tells us beekeepers that something is wrong in the colony with the queen. They might be sick. And so we have to do a little bit of investigation for that. And so how do antibiotics affect the gut microbiome of honeybees? Well, I mentioned before that it has been noted to impact the gut microbiome. And again, the antibiotic tetracycline or terramycin has been used in beekeeping for over 50 years to help combat against these bacterial brood infections. So we know in humans, these persistent infections with antibiotic treatments following can increase a lot of resistance in the United States especially. And we know that antibiotics are exactly that. They're an antibiotic. They're meant to kill bacteria. And so it will have some sort of disruption on the microbiome, especially in the gut. And so we've noticed that as tetracycline has gained resistance in the United States, can help the community adapt to the treatment, but also spread resistance to the pathogens that might be inside of that gut community too. And we have several strains or genetic variants of a species. I'll define that a little bit more later if you're confused, but several strains of causative agents of American foul brood have acquired tetracycline resistance. And so we really don't know how antibiotic treatments impact the microbiome of honeybees. And so what the study I'm going to talk to you about really wanted to look at is how the microbiome size and composition is going to change over time when exposed to antibiotics. What happens to bees when they're fed antibiotics, and what happens when they're fed antibiotics and if the infection of opportunistic pathogens happens. Okay, so first and foremost, what we do when we're trying to look at how tetracycline or how a chemical can impact the hive or microbiome of honeybees, we will collect hundreds of workers from the colony. We will actually paint their backs with a color, like a paint marker. It's extremely time intensive, but really fun. We put them into cup cages and we'll give them the tetracycline, or we won't give them the tetracycline as a control. And then we put them back into the colony and sample them at different time points. And this was done in a 2017 study by my graduate advisor. And so we can use that sequencing technology and use other types of technologies in the lab to understand which bacteria are present in the gut, which bacteria are not present, and how those abundances have changed over time after given tetracycline. And so truth be told, tetracycline does alter the microbiome diversity of honeybees. So we have these two different diversity metrics that we use a lot in the field, and one of them is called alpha diversity. Alpha diversity simply means how many bacterial species are in an ecosystem. And so what you can see here is that alpha diversity is reducing the amount of species in the bee gut. And this graph here on the left, or excuse me, the right side shows on the y-index alpha diversity, and on the x-axis you have a control versus treatment. So on day 0, there's really no difference, nothing really happened. But on day 3, you can see that the number of bacterial species was dramatically reduced in the bees that got the tetracycline treatment. And you can see those similar trends in days 5 and 7. Well, another diversity metric that we look at is beta diversity, which is how does one microbiome sample differ from another microbiome sample? And you're really trying to see how similar or different two microbiomes are compared to each other. So tetracycline causes these gut microbiomes of honeybees to actually become less similar. And so that means that one microbiome is either gaining or losing species, and another is gaining or losing species, but they might not be the same anymore. And so you can see that, again, on the y-axis, we have the beta diversity metric, and on the x-axis, you have control and treatment. And you can see there's dramatic differences, meaning these microbiome communities are becoming way less similar to each other because they're given tetracycline. All right. So another thing we want to look at is, again, just a different way to see that same data. I have a lot of fancy jargon up here, but really what I want you to take away from this is that these graphs are showing you the different treatment days for the treated bees and the control on different days. And the control bees are in blue and the treated bees are in red. And really what you're seeing is that the control bees are starting to cluster together in these graphs, and the treated bees are also clustering together, which again really shows us that tetracycline has a really big impact on the microbiome diversity of bees, and this could be potentially harmful for their health overall. All right, and lastly, what I want to show you from this study is that you can see the actual differences in abundance of bacteria. And so what you're looking at here are relative abundances and absolute abundance. And so on the y-axis, you're looking at percent of bacteria. On the x-axis, you're looking at different sample numbers, sample 1, 2, etc. And so by doing this type of sequencing, which we call 16S, it provides us kind of a relative abundance of how much bacteria is inside of the bee gut and why that's important. And for absolute abundance, we use another type of technology in our lab to get an absolute count of how many bacteria are present. So you can see that sample 6 and 7, for example, they seem pretty similar. They don't seem like they're too far off from each other and have really similar amounts of bacteria in that gut. But if you do it in a more absolute or exact count, you can see that there's a huge difference between how much of that gut bacteria is present. And what this gives us is a more accurate description of how that community is structured overall. All right, so the conclusions from this is that tetracycline is harmful to honeybees, and it can decrease their survivorship overall, and it can also alter that gut microbiome. And what this paper also found is that when that microbiome is altered, it's opening up the honeybee to be more exposed to opportunistic pathogens that could harm them overall. All right, in the last part of my talk today, I want to talk about current projects that I'm working on. So we talked earlier about factors that could influence a microbial community. And these could be things like the diet you have, how old you are, your genetic background, your lifestyle, your location in the world, as well as how much microbes you might inherit from your mom or from horizontal inheritance. But nearly everything has some correlation with having an impact on the gut microbiome, whether that be a probiotic yogurt or eating the right foods. And so factors of influence of diet have been shown through many different studies of humans that having a low versus high fat diet can alter your microbiome composition. It can also be said that native peoples, the Inuit microbiome, can change over seasons because of how different their diet can vary across seasonal lines. In addition, your age is going to really impact your microbiome. So we can see that as you age, you're going to increase or decrease your alpha and beta diversity, as we just talked about. And this is mainly going to be because your gut architecture is physically changing as you age, which is pretty cool to me. And so this is mainly going to be due to your lifestyle, where you're located in the world, and your diet, but age does have an influence on your microbiome composition. But what I'm really trying to understand with my research is how honeybees are going to have different microbiome compositions because of these different factors. And so specifically, I'm looking at how diet, the honeybee age, the genetic background of the bees, and their job that they play inside of the colony could have an impact on what their gut bacteria could look like. And so although honeybees aren't eating cheeseburgers and avocados and do not resemble a human baby, they are foraging from places like this. This is a monocrop of almonds in the Central Valley of California, and there's very minimal diversity here. They also are going to age over time, and so they're not always going to be a tiny little honeybee egg or this squirmy little larvae here. They're going to be full-fledged bees at some point. And how does that maybe impact their bee gut bacteria? They're also going to have different genetics in the colony, which gives them maybe different types of bacteria in their bee gut. We don't know that yet. And also they're going to have very different roles inside of the colony. So this bee here is going to be feeding the young, and she's called a nurse bee. So many different bees play different roles in the colony, and overall, that is going to potentially impact their microbiome composition. So with that, I wanted to just give some cool resources if you're interested in what I just talked about, or if you think that maybe honeybees are cool, but I want to learn more about microbes and how they interact with the world. I really suggest reading this book by Ed Yong. It's incredible, and it taught me a lot about this field, or this book here by Rob Dunn. And I'll share these slides with anybody who wants them afterwards. But with that, I really thank you for your time and your attention. Thank you, Morehead, for allowing me to be here. And this is my lab, and thank you to the bees. And that's it.
All right. Well, so we can give time for folks to put some questions in the Q&A. I have one that I can get us kicked off with. So you did say that it's not good for biodiversity to have when they just like go for one type of flower, like the almond orchard or whatever. And that can like drastically alter the taste of honey. Like an orange blossom honey is going to taste a lot different than like a wildflower honey. Does it taste different if they have been treated with the antibiotic?
So I can't say. That's a great question. Yeah, first and foremost, you're definitely right. Honey does taste different depending on what they forage from. Absolutely. There are very, very specific guidelines to when you can treat your hive with antibiotics, and you cannot treat during a honey flow season when they're actively foraging. Therefore, they're not going to be treated with antibiotics as well as collecting nectar for honey and therefore potentially exposing it to humans. You cannot do that. So there are very specific regulations. There have been trace amounts of residues of antibiotics that are not directly antibiotic. It's like a secondary metabolite found in honey, and that's been shown through previous research. However, you won't necessarily, I don't know about taste differences, maybe for honey. Yeah.
Okay. Well, thank you.
Yeah, no problem.
We have the question, what can we do on an individual and societal level to help protect the bees?
Yeah, that's a great question. I think it's one of my favorite questions too, because there's actually a lot you can do. So first, I would say if you have access to green space or a yard or anywhere really, definitely think about planting some native flowers. Bees are really hungry right now. North Carolina is going through what we call a dearth period. And what that just means is that there's not a lot of nectar around. And so if you have a really nice patch of flowers in your yard, you'll see tons of bees coming around and they're just saying thank you. You just can't really understand it. So definitely plant some flowers. And if you're really interested in bees too, there's tons of resources out there for you to just become more knowledgeable about them. This might surprise folks, but wasps are indeed pollinators. And although scary and a little bit more aggressive than honeybees, they're also really important too. So you can actually do a lot to become just educated about different pollinators, as well as planting flowers for them in your backyard. Yeah, great question.
So yeah, I actually planted a bunch, but the deer keep coming and lopping them right off. Like, man, my gardening is just hitting obstacle after obstacle.
Yeah, I feel that pain. Absolutely.
Are you keeping any bees now, or do you have any intent to keep bees now?
So I have bees for my lab, clearly. So I have, let's see, probably 6 or 7 colonies for my own personal research. I don't have any personal colonies right now. I think there's such a huge difference between doing research on bees as a job and then having them as a personal kind of pet, if you will. So no, I don't have any right now, but I hope to someday.
Okay.
Yeah.
And does anybody have any other questions? Because we are about out of time. All right. Well, then we'll go ahead and conclude for the evening. Megan, thanks so much for joining us today.
Yeah.
Thank you so much, everybody, for all the help that you've given the Teen Science Cafe as we've pivoted to virtual. Youngko, Riley, Kim on the back end doing the webinar that nobody can ever see, and then a lot of our consistent attendees, Ava and Violet. Thanks for continuing to grace us with your presence, and we just hope to see you all in person in the fall. But fingers crossed, we'll have to play that by ear. So without dragging that on for too long, I bid you all a good evening, and I hope you have a good, lovely summer. Thanks again.